of diagnosis that are available. I would want audio. Yes. In diagnosis of COVID, what do you know about the types of diagnosis that are available? There are two types of tests, right? One of it is, uh, we should say PCR based. We shouldn't say PCR mediated, but that's all right. Yeah, one of it is rapid test, and the other one is RT-PCR, RT-PCR based. So that's right. The rapid test is the based on detecting the antigen, right? And the other one, the RT-PCR based one, based is dependent on the qPCR or real-time PCR, the same thing. And in these two, we have that is where molecular techniques or genetic engineering or recombinant DNA based ones, or you can say the molecular diagnostic kits come in handy. So what I would suggest is, I think I must have told you this in the previous or the last class as well. Try to know about the principles of rapid test, rapid antigen test, and the RT-PCR test, how it is done for COVID. From you should have started with. You might actually want to note it down so that you can look into those things and see if you can gather enough information. In the last PPT that I have uploaded, I did try to put in some pictures, okay, uh, which might be which might give you an introduction into the techniques. So from sample collection, this is a uh, real time application, right? So from sample collection, the processing of next one would be processing samples, and then the test itself, and then how one would interpret the results. I want you to know about the when you are trying to do these things, sample collection, processing of sample, and the test itself. You would also want to know about the design. Say in case of rapid antigen test, there is a strip. We should know how it is designed. In tomorrow's class, I hope you will try to find it out if you didn't find it yet. Similarly, for RT-PCR, the key will be about understanding the principles of RT-PCR. How does it work? What is CT value? How can one tell which one is positive and which one is negative? And first thing, why is it called real-time PCR? And how is it different from traditional PCR? So there are also more things in RT-PCR, like the ways of detecting the product after each round of PCR cycle. That is real-time analysis. How is it happening? Cyber green. We might also have TACMAN probes and other things. We have learned it before. But in previous classes, we have learned techniques as such. But eventually, it will become onto you about how you use it in real life situations. In this current situations, COVID is the issue, and we all have to try to apply our knowledge towards that. At least I would put it this way: you already know many of the techniques, or at least they have been taught to you. You have an introduction at least, and it is much easier. It's very much easy for you to understand the principles, and so you shouldn't hold back in trying to know the principles of diagnosis that are used in COVID at least. The second one is about vaccination. Vaccination is a preventive measure. It is somehow making an individual already resistant to uh, the virus. In vaccination, we can do many new things, and I hope you know about the story of the person. I forgot the name. Sorry, but that lady, okay, she has been working hard for a long time. She hasn't been able to generate funds and so on, and the institute almost fired her. But eventually, they gave her an option: either you leave or accept a demotion. Okay, so coming from, I think, from a professor, she was supposed to become a professor. She was at the level of associate professor, and then probably she was demoted to assistant professor or something. I can't recollect exactly. She, because she has her own commitments, she took up, she accepted for the demotion. She continued working on it, even though it was very difficult because she was doing a lot of basic science. Basic science is trying to, and I mean, some science that not everybody here at that place probably, or she couldn't generate funding. So now the the vaccines that were based on mRNA, the majority of information comes from her work. I mean, not, there are many other people also who did that, but this is an interesting story how one woman held on to her job and research goals and eventually succeeded despite the problems that she had, uh, probably at family burden and also at the profession. Despite that, she has done. It must be a tough job. So we have learned about subunit vaccines, where we just give the subunit protein, or we can give DNA. We have learned about DNA vaccines, where we give the DNA. DNA gets integrated, hopefully gets integrated into some of the cells, and then they produce the gene is expressed, and they produce the protein, and uh, that protein, the immune system develops antibodies against that particular viral protein, so that the memory is continuously there, right? Subunit vaccine, directly you express, you produce the protein outside the cell, and then uh, you give it to the individual as a protein. uh, for in DNA and also mRNA, how we produce these things is dependent on constructing a recombinant DNA. And whether to give a protein or a DNA or mRNA is a big question. There are advantages and disadvantages to each of these. Therefore, I would like that you people are able to look into them. And what types of vaccines? Again, in the last PPT that I have sent you, majority of the details are already given in there. I hope you will be able to have a look at that. So that is with regard to COVID. And there is one more thing I would also want to add. People are saying there is, I mean, there are from UK and other things, there is a second, or it is this COVID-19 virus is mutating. And one of the strains in UK apparently is much more contagious. And if we are getting a vaccine for one strain, is it can it provide immunity to the second type of the new strain? Those are some of the other questions that we should also think about. I think we have had a small discussion on that at some point, not exactly based on COVID, but these things. So for tomorrow, I think it will be a good idea if you can look at the COVID situation in these slides. Try to know the diagnostic principles that are being employed. Which one is better? Always try to have advantages and disadvantages or limitations of each of the techniques or strategies. Rapid test has an advantage, but it also has a disadvantage. RT-PCR has an advantage, but it also has some limitations. So we should try to figure out what to do the way we learn about these things. Okay. And I was hoping when in the WhatsApp group, I asked for any list several times, but I didn't get any response there. If you have already sent it, I could have or put it up now itself. Uh, I'm reading a statement. One second. What is it? Uh, I actually don't have a clear-cut idea. I haven't studied this strain yet. And if I want to know, I should know about the. So I hope you all are reading the chat here. Shruti says, "What do you think about Pfizer's statement that taking the vaccine twice can safeguard against the new strain as well?" First things, I mean, if I keep answering this, then I will give away what I wanted to tell, wanted you to look up. But I'll let me try to speak in. First things, I'll only give more questions, which will eventually address this answer. First thing we need to know is if they're giving mRNA, for which gene, for which, what does that mRNA code for? Mostly it is about spike protein. And so if they're saying it in two doses, is it for the same gene, same protein, or is it for different proteins? Is second question. Say if it is for the same protein, and then it is unlikely to make big difference. Uh, but if they are giving for two different proteins, say first shot for first dose for one and second dose for a different protein, that I don't think is what they are doing. I think they are giving a. It is second one is like a booster dose. I think I haven't gone through each. There are two or three more companies putting up. The principles will remain will depend on that. But what, to my knowledge, about the second strain is, I think when you give a protein and the protein is expressed, when you give an mRNA and it produces a protein, it's unlikely that all parts of the protein are going to get mutated. If that is the case, that protein will lose its, it will not have a proper structure. And if it is not structurally correct, then it doesn't perform its function to give stability to the virus. So while there will be mutations, it's unlikely that there, say for example, what do you expect? If a protein is given, will that protein generate only one type of antibodies? I mean, antibodies specific to only one epitope, or antibodies specific to multiple epitopes on that protein? What do you think will be the answer? A protein can consist multiple epitopes. So even if the mutated strain may lose or change one or two of the epitopes, there will be other epitopes that might be still relevant, to which, if vaccinated, the individual must have had antibodies against, and they will probably provide immunity, good enough immunity. Did I answer some questions at least, Shruti? Maybe I will also try to discuss this in the next class. Uh, okay. Pratna has a question. That's while while selecting the protein for vaccination, won't they look for a protein that won't mutate? I think the question should be, every protein, every gene is prone to mutation. Everything is everything, anything, mutation or variation in evolution is random. Anything can happen anywhere. But you have to we have to think of it like this. If a particular if there are two proteins, say protein A and protein B, if protein B is not so essential for the structure and stability of the virus, even if there is a mutation in protein B, the virus can still exist. But if, say, protein A is very important for the formation of the envelope on something like that, say, an essential function, and even slight mutation of it may cause it dysfunctional. And if it becomes dysfunctional, virus doesn't exist, right? So the mutations can be a gain of function, neutral, or loss of function. And any gene is prone to mutation. So for with regard to we have to look into the essentiality of the protein. And the second thing is it should be exposed on the surface, hopefully. That is when it will be accessible to the antibodies. The third will be that if it has multiple epitopes, it is better. Okay. Uh, yes. I hope I answered a little bit of it. But yeah, there is nothing like a protein that won't mutate. All will mutate. There is only thing is after mutation whether it will exist. I mean, is it a gain of function, loss of function, or neutral? That is the only matter that changes. Any other questions? Yes. So when is the exam? RDN exam?
It's on 28th, sir.
Okay, 28th. What are you doing for gate? Any ideas? So in okay, I hope you're doing well for the gate. RDNA, many of the things that are there will usually come handy, especially in analytical questions. And if you look at, and if you, I have sent you, I have posted on December 1st some material. That is, I made a material for I had a coronavirus based lecture sometime recorded a long time ago. That is one thing there. But I also had a PPT that I posted and some YouTube videos as well. I hope you have watched them and they will be very important for your learning. There is also one Word document I posted on November 30th about discussion on and some question papers. Hmm. So if you look at the table that is there, I have given a sample question as well. Okay. So all the if you have gone through this particular document, you have covered all the question paper. Okay. uh, I have given you what you have to look for about what. Okay. And then I've also written how you are supposed to, sorry, how you are supposed to present the, one second, sorry, how you are supposed to present the answer. Please download the document, read through the learning outcomes, and then also about several important topics, how you can apply the principles or techniques that you have learned in this course for various questions. And if you see there is a sample question that is also given with absolutely similar wording as well. So that you will probably know how to expect. You can actually, looking at the table, you can make your own questions based on that. You have that much information is already given to you, so there is nothing that you would actually you have everything basically. So I hope you can try to do those things. Okay. What other works do I have? I have to send old question papers. That is one thing. uh, is there anything else that is missing? If you tell me, I will try to make up that stuff. Okay, while you are, yes, please, somebody. Yes, somebody was about to say something. No. I am unable to find the DNA course here. I mean, question papers in they said that is already uploaded, but I can't see it. Just stay online 1 second, please. Okay, I'm going to I'm going to say send the question paper now. One word question paper. We'll have a quick discussion of it. I think that's better. Oh yeah. I also remember I actually made you all to solve CA question papers, right? The the question papers will be exactly the end semester is also is almost the same as what I was telling you. So you can I will try to post. Please go on to your material in Google Classroom, and then um, I'm posting it now. Yes, it is posted on the it is posted on the Google Classroom. You can please download it, and we will have a quick discussion on that. Sorry. Can you let me know if it is visible in the Google Classroom as a material? Yeah, it must be.
Yes, sir.
Okay. If not, you can also see it on my screen. Uh, the model is the same. That is, you'll have part A and part B. In part A, you have 6 questions, out of which you can answer any four. And part B will have 1 question, and all the questions are 20 marks each. So each question can or will have, actually, most of them will have sub questions. Yes, I will read the question. I hope you can see it anyway. A researcher is using lambda phage based vector, lambda EMBL3A, for the cloning of several fragments of bacterial genomic DNA. The researcher wishes to clone 20 kb fragments to in lambda EMBL3A. Discuss at least 2 strategies through which plaques are formed only by the recombinant EMBL3A. The question is general, but the second part of the question is actually highly specific. In this particular question, in the first one, you will say about the structure of EMBL3A. You'll have about left arm, right arm, and the stuffer region. You would also describe what is the significance of left arm, right arm, about the essential genes that are involved in replication or structural proteins for formation of lambda virus. And you should also tell about if it is an insertional vector or it is a replacement vector. EMBL3A is a replacement vector. And the stuffer region, on the bordering the stuffer region, we have 3 different restriction sites in particular order: EBS, you can say, EcoR1, damage 1, and SAL1 on both the sides. So why should there be 3 restriction endonucleases? Why not just one? And you should also be able to tell about in the stuffer region if there is red and GAM. So we know we will digest the genomic DNA. We will. Uh, there is one more critical aspect that we should be discussed. That is about treating with alkaline phosphatase. What should you treat with alkaline phosphatase? Is it the genomic DNA or is it the lambda phage vectors? Typically, vector is the one that we treat with alkaline phosphatase to. And why do we do that? It is to prevent religation of the vector, because it will give false positives or kind of things. It will form clones and it's unnecessary mess for us. In the case of making genomic libraries, what we are bothered about is losing the integrity of the genome. When we want to analyze it, we want it to be the correct proper order. So we don't want different genomic DNA molecules ligating, because it will show us it'll get cloned in wrong ways, and that is a problem for us. So how do we prevent, if we are going to use alkaline phosphatase for the genomic DNA fragments, how can we prevent false positives? That is where we are going to use 2 steps. One is by using 2 different restriction endonucleases, and then removing the smallest fragments using isopropanol precipitation. The larger fragments, they will precipitate faster, and the smaller fragments that are formed in between, they will not. Right. I hope you can recollect the things. I can have, if I had known, we would have discussed that. But I want you to recollect. Every time we see and learn is different. Often you try to recollect what you have learned, and that makes a basis into your mind, and it will increase your memory on that particular concept. Whichever memory works this way, whichever uh, moment, person, or a logic, concept, whichever thing that you recollect often in your mind is what that is how you create a memory of. Okay, so try to. I could have shown you the PPT and then just said, yeah, this what is what happens and happens. Instead, try to recall in your minds. You have, I am sure, from the group that I can see, you all have followed well, and I'm sure you can just try to recollect from your visualizations if you had any, at least the terms that I'm saying, and then when you look up the answer you'll feel much comfortable with it, and after a while you can't forget it even if you want to. That's how strong recalling makes it. So try to recollect what you have learned, and that's always a good thing. So yeah, with this, there is also one more thing that you need to do is to how to prevent false positives or vector religation or uh, unwanted plaques to form is using spy phenotype. That is by red and gam genes. We should use P2 lysogen as a host. We should not use E. coli. Spy phenotype only works with regard to P2 lysogen. Yes. So if there is red and gam, uh, what is spy sensitivity to P2 lysogen? Right. If there is red and gam, it does not form in the lambda phage. It does not form plaques on P2 lysogen. If there is no red and gam genes, which are actually present on the stuffer region, which, if replaced by the genomic DNA, will form plaques. If it contains stuffer region, then it does not form plaques. Yes. So the second question is about PUC 18. It's a this question paper is actually kind of straightforward in many aspects, but still it's about how you represent and the critical details that you speak of. Please remember this, that don't make trivial sentences. Anything that you write and depict in all of the answers, you should encourage, you should write your using flowcharts, diagrams, and so on. But the critical note should be given, and why that particular step is important. The logic of it should be evident to all of us, right? That is when we know how the depth of your content. So please focus on trying to draw it, write it out in a on a paper. Also remember this, that when you keep looking at the diagram and you haven't ever drawn it before, then it's going to be really difficult for you to draw it in the exam paper the first time, right? It is always a good idea at least to have 3 or 4 practice, if not for all the questions, at least for some questions you should have good practice so that you know how to present it. Here, when you are presenting it, it should be clear to us. You should be specific. You should have labeled everything, and you should have given the logics of why that particular event is done, or a factor is present or component is present. What is the use of it? That should you should be able to describe it. In EMBL3, the first question, I think the minimum number will be around 2 pages, or at the most 3 pages, and all of it will be, majority of it will be in diagrams. That gives 20 marks. It's not that you have to write, if it is 20 marks, you have to write 5, 7, or 8, 10 pages. It's about how well you write, how how many specific details you write. That is the point of evaluation. In PUC 18, uh, it has two things that you have to know: the design, ampicillin resistance. There are unique restriction sites. There is also for blue-white screening, which is based on alpha complementation. I hope you remember alpha complementation. If you're going to use PUC18 or anything that is based on alpha complementation, you should remember what type of host you have to use. You have to use M15 based E. coli, not the wild type, because wild type has laxate, functional laxate gene, and therefore it will give blue color irrespective of whether the plasmid is present or not. So those kinds, and also if you're going to use it, you have to ensure that the host strain is antibiotic, I mean ampicillin sensitive, right? That is where the selection actually, that is how you'll be, your selection thing will come handy. So keep those two things in your mind about it. Yes, blue white screen, you will have to depict a answer and say why, what kind of cells, transformants or non-transformants, what kind of uh, colonies color they would they produce, why would they produce. That is where you will be describing more about alpha complementation. Second, the third question is gene therapy based strategy for treatment of type 1 diabetes with justification for the choice of cells, approach, and the gene construct. Based on completely on the paper in fourth unit about engineering mouse enteroendocrine cells, and that is where this question has specific specificities that you should say. Uh, Justification choice of cells. Should we take beta cells? Beta cells are already non-existent in type 1 diabetes is based on insulin deficiency. The insulin producing beta cells are killed mostly. It could be because of viral infection, some type of viral infection, or it could be because of autoimmunity, or there are many other reasons as the cases may be, and sometimes we don't know the reason too. So in that case, it's unlikely we are not going to get beta cells. So what else can be done? You have to look into that paper. Intro, sorry, enteroendocrine cells were there. They, why is that a cell choice? Because it responds to the glucose in the blood, and it has all the proteases required for processing insulin. So you should know about how, you should talk about how insulin is processed and secreted, what are the requirements for it. You should discuss about what is type 1 diabetes in the first place. Then we should also discuss about sensing the blood glucose. And the third is to release the insulin on time based. And if this can be done, then there will always also be limitations, right? And you have to talk about those limitations as well. Uh, the third one is, sorry, the fourth, I will, I think it's 50, differential screening and subtractive hybridization. I hope we have discussed about, we did discuss about differential screening, and did we discuss about mRNA hybridization on the plate, labeled RNA, cDNA hybridization? If not, we will try to take it up. But subtractive hybridization, I can remember that we have discussed clearly. There is a tester and there is a driver. You have to do it A minus B. Whatever is resultant is present in. So subtractive hybridization is something like this. You are going to tell which genes are present in cell type A, but not in cell type B. So all those genes that you will be able to clone after subtractive hybridization are those genes that are influenced by the by a treatment or whatever it is. Okay, I would want you to look at write about those specific details. It's always easier and better for you to take up as an example, like I said, cell type A and cell type B. Assume these were grown in same cells in different, grown in treated or untreated, and then you can build up the story and write an interpretation and the results to it. That's a good way of presenting your knowledge on that. Compare the principles of East 1 hybrid system and 2 hybrid system. I think we have done it well. I will leave it. I will just ask this question: what is the use of East 1 hybrid system? Anybody? I don't. I was trying to not ask you people any questions because as soon as I start asking questions, you might run away. I already have only 14 people. So, anybody? Even if it is wrong, it's all right. Just
To
say. Yeah,
determine the interaction of protein with the DNA.
that's right. Yes. Why is it called one hybrid system? Anybody? How many hybrids are we making in this type of things? What hybrid is it, first of all? It is a hybrid of, it's a fusion protein that we are producing. That is of binding domain of gal4. No, no, no. Activation domain of gal4 and the protein that we are testing, which might bind to DNA of our interest, right? That is a bait sequence. So you can have a look at that. Because we are making one hybrid, using one hybrid, you call it as one hybrid system. And as Hrithika said, it is used for to find out if there is an interaction between a protein and DNA, and a specific DNA. The second one is two hybrid system, and what is its function? Or what is its application? Why do
Protein
we use
protein
it for?
interaction.
That's
Protein
right.
protein interaction.
Yes, yes, you're right. If you want to find if there is an interaction between a protein and another protein, then you would try to find that. For in this case, we will need to make two hybrids. That is one protein with fused with the activation domain, another protein fused with a DNA binding domain of gal4 transcription factor. You have to remember those specific details. Yes. Uh, yeah, that will give. So when we have the questions, usually the questions could be, uh, you can make a choice, right? 6 questions. Out of them, some will be critical, but you don't have to write too much if you know the answer. Some will be simple, but you have to write fast. You should be good with. I would like that somebody is good with. Where you know the answer, you can figure out the answer and write lesser. That's always nicer, but it's up to you. It doesn't. It's all right. So this is a whole scenario. Which, if we were to the fifth question, and I think in one of the question papers, in I made you do for to write for CAA question papers. This question must have come, and I hope you all have done it well. Okay, so I'm not going to discuss this at this time. Similarly, did you people get this also in CA question paper somewhere? Can anybody tell me? It is about making a probe for northern blotting or southern blotting.
Yes, sir, we got it.
You have done it as one CA, right?
Yes, sir.
Yes. Okay. This one, was this question there anywhere?
Yes,
Uh,
sir. This was also there.
this is also there, right? Yeah, I picked those the same things, but I have to say, majority of the people's answer is not to the mark. There I gave benefit of doubt to you people, but maybe we'll have to discuss this tomorrow, maybe. You are required to identify 5' end of mRNA of a bacterial DNA. Design a methodology that you would employ in fulfilling the objective. What could be the answer for this question? 5' end. 5 prime leader region, the start transcriptional start site of mRNA. What technique would we use? Anybody?
Sir, use the we could start from the 3 prime end using a poly A tail. Recognize a
Yes.
poly A tail using a primer and then carry out PCR.
Yes. How would you identify the sequence? Or there is a name of the technique, is there? Simple name. It's almost what you said.
Sir, I forgot the name.
Okay, that's all right. It's primer extension. Primer extension along with sequencing reaction, if you do, you can figure out the transcriptional start site of the mRNA. Okay, we have discussed this in one of the lectures. Yes, here the question is not telling you the technique name. We are telling you the need, and you are supposed to figure out the technique. Say, as Pratna said, you don't have to really remember the name. Also, you can actually try to find out your own logic and use it. And if the logic is valid enough, you would get marks for it. Okay, always keep that in mind. Also, if you didn't know anything, write something that you think is correct at that moment. And as I always believe, I know that I also use this phrase, right? Is something better than nothing, or nothing is better than nonsense? Something is always better than nothing. That is the goal with biology works, and it can. It's like something is better than nothing is like the hope. So we always have to be hopeful. So the second question is explain the principles and the components required of the techniques, procedures, labeling that you would need to use in this case. So for that, you will need to, for primer extension, you will need to have a primer. You should have uh, labeled the primer. You should have used sequencing reaction, dideoxy method, Sanger's method, right? And then you should have performed the experiment and then written it. So first 5 marks is just basically for you to tell about how you would want to go or the name of the technique. The next 15 marks is actually for the rest of the things. Okay. I'll stop here, and I would quickly want you to tell me what all things should we do over the course of 3 or next 4 5 classes. First thing is, say this question paper is already over. I don't want to discuss this. I'll see if there are any more old question papers with me, because these are the only ones that are uploaded actually. You already told me what is that? CRISPR is one thing. What else? Can I make a note? Genomic libraries is one. EMBL, or what is that? Shubhashri, you told me.
Race
Sir,
PCR, sir.
lambda chapter.
Lambda carry on. Lambda Cheron or
Yes,
something. Yes, yes.
yes, sir.
Okay. Yes. Somebody else? What else did you say? Uh,
Race PCR, sir.
yes, race. Yeah. Race PCR. Race is just for identifying the ends, okay? Amplifying the ends. So just keep in mind about it. Yes. Anybody else has any more?
Sir, there's a particular topic in epigenetics where you explain how the modifications are transferred to the next generation. I did not understand that cycle, sir.
Is it genomic imprinting
There
or?
was yes, sir. In genomic imprinting.
Okay, but I don't think that is important for you, but I'll try. Genomic imprinting. When you say from one generation to another generation or one cell to
So
another.
there was an image with the cycle, how it gets erased, but then it gets the genomic imprinting comes back after the hybridization.
Okay, just send me that image or so that I'll be clear on what you exactly wanted. But otherwise, I written it as genomic imprinting.
Okay, sir.
Yeah, but remind me, just post a picture or something of it to WhatsApp.
Okay,
Anything
sir.
else? Anybody? Yes. Uh, okay. In general, I'll ask again. Has there been any preparation at all so far, or not much? Okay, good. Prepare well. Don't have any other types of ideas in general about exams. Things will happen, not happen, or something. It doesn't matter, whatever it is. If you're preparing, it is useful to you. Keep in mind, just go ahead with that. Say, gate will happen, right? It will be useful. Everything is useful. Otherwise, there will be a lot of waste. There will be time that is lot of lot of time that will go waste. So just keep that in mind. Yeah. Uh, okay. There is. Is it okay to ask for topics that are not too important? That's difficult to answer. I actually gave you the questions that are very important. So if you know those, I don't think I should say anything about that are not too important. But because you can come back at me and say you said it is not important, then it will become a difficulty. So choice is there. Whatever it is, you have to use your choice in that level. Selective reading is not the best, but I already gave you some important. First thing is cover them up. Try to know and plan your answer properly. Practice at least 2 or 3 answers. Have it in mind how you would do it. Don't assume that all the figures will come to your hand right away. At that point, it's not just about drawing, right? It's also about presenting it. So learn or at least practice at least 2 or 3 answers to present them. Then you will get an idea about how you want to present it, and that will be good enough. If you want, you can send it to me beforehand now, while this in this whole week, and see for this question, is this good enough an answer? That's also fine with me. Okay. My strategy is simple. I will try to uh, make you do stuff. I wouldn't do anything that will stop you doing from stuff. Yes, you doing is the essence of all education, not we teaching. You doing is important. Okay, then we'll see you tomorrow, and I'll try to cover some of these topics, and I'll see if I can get any other old question papers. Okay, I'm very happy that you people turned up. Uh, thank you for turning up. Okay, see you tomorrow. Bye. Yeah, bye bye.
Thank you, sir.